It's time to get started. Again, hello everyone. Greetings of the day on behalf of the Bioprotocol webinars team. This is Antara Sengupta welcoming you to our webinar series, New Tools of the Trade in Life Sciences, Technologies that Enable Breakthroughs. Bioprotocol is an open access, peer-reviewed online journal dedicated to making research more transparent and reproducible. We sincerely thank you all for tuning in from various locations and being part of our mission. Today's webinar explores OrigamiFISH, a universal label-free single molecule method to visualize DNA origami nanodevices across biological samples. We're delighted to have Dr. Wendy Wang with us to share her expertise on this fascinating topic. Dr. Wang, thank you for graciously accepting our invitation. To moderate today's session, we have Bioprotocol Webinars Program Lead, Dr. Renee Weisbauer with us. So without further ado, Renee, I hand it over to you now. Thank you.
Okay, well, thank you, Antara. All right, it is my distinct pleasure to introduce today's webinar speaker, Dr. Wendy Wang, who is currently a Damon Runnan postdoctoral speaker, Dr. Xiao Wang's lab at the Broad Institute in Boston, where she works on developing spatially resolved approaches for mapping neuronal connections. Wendy completed her PhD in molecular genetics at the University of Toronto in 2022, where she researched neural circuit formation, focusing on neuronal morphology, connectivity, and cell surface receptors. And then following her PhD, Dr. Wang worked briefly with Dr. Leo Chu's lab at the University of Toronto on DNA-based nanotechnologies, resulting in the development of OrigamiFISH, a label-free and universal method for the single-cell molecule fluorescence detection of DNA origami nanostructures in cells and tissues, which she's going to talk about today. Wendy also won numerous awards, including the BrainStar Award from the Canadian Society of Neuroscience for her work developing a multimodal diversification map of cerebral molecular layer interneurons to better understand complex brain circuit assembly. And with that, Wendy, the floor is all yours.
Okay, great. Thank you so much, Renee and Antara, for the really kind introduction. As Renee mentioned, my name is Wendy, and I'm really excited to be here today to talk about some of the work that we did when I was a postdoc at the University of Toronto in Dr. Leo Chu's lab, where we developed this method, which we call OrigamiFISH, which allows for the visualization of different types of DNA nanostructures across various cellular and tissue model systems. So just as a brief overview of the presentation first, I'll start with a little bit of introduction onto the sort of general concept of these types of DNA-based nanostructures for nanotechnology platforms, as well as their various applications. I'll then go into sort of some of the current approaches that people have used for detecting them and visualizing them, as well as their limitations and how those sort of led us to initially propose and develop OrigamiFISH as an approach, as well as the validation and applications that we've applied OrigamiFISH towards. Okay, so just to get some basics out of the way first, the star of the show today is definitely DNA. So as I'm sure everyone here already knows, DNA is our genetic material, right? That's present in all of our cells. So for the sake of this presentation, DNA is a nucleic acid, which is double-stranded. So it's in the form of a double helix that then encodes for our genetic information. And so for the field of DNA nanotechnology, then we essentially wondered as a field whether we can, in addition to thinking of DNA as the genetic material, also kind of repurpose them as a synthetic building material of sorts. So can we actually make things out of DNA, which is useful for different types of biomedical applications? So what I mean by that is we have different approaches, DNA origami being a main approach that we adapt in the lab, which is a method which allows us to essentially fold really long pieces of DNA into different types of nanoscale shapes. So just like how in regular paper origami, you can sort of fold a piece of paper into a variety of different shapes. Here we're folding pieces of DNA. So typically in DNA origami, we have a long strand of single-stranded DNA, which we call a scaffold, that's shown in cyan on this slide. And this is typically a single-stranded bacteriophage genome, so approximately 7 to 8 kb in length. And then because we know the sequence of this sort of DNA, this long scaffold DNA, based on that sequence alone, we can then design sort of hundreds of these shorter synthetic oligonucleotides, which we call staples, that are shown in orange on this slide, which are essentially programmed to come in and pinch that long strand into any shape that we would like. So in this case, these staples were programmed to form into a rectangle and shape. And for the rest of my presentation, I'll sometimes be calling these nanostructures as nanostructures, or sometimes just referred to as DNs, just for brevity. So fortunately, we're not also limited really to creating those, really boring 2D rectangles. These are just a few other examples of different shapes that ourselves and others have made over the years using the same DNA origami approach, using the same scaffold strand. So we have 2D shapes at the top, like the 2D rectangle I just showed, as well as these smiley faces, as well as 3D shapes like this barrel here at the bottom. And just to make sure they're actually forming, we can also use imaging approaches, so typically electron microscopy images, which you see on the right of each animation, which are actual structures made completely out of DNA, which are confirmed to have folded in a test tube. Right, so just as a summary then, DNA origami is a really versatile approach, which really allows us to create a number of different really diverse shapes, which are completely self-assembled and made completely out of DNA. So that's great that we can make shapes, but why would we want to do this, right? What's the actual advantage of doing things this way instead of making other types of nanoparticles, which, like, for example, gold nanoparticles or lipid nanoparticles, which are a little bit more common. And as it turns out, the same sort of features which allow us to make these types of DNs also give them a number of advantages when we're thinking of them as a nanoparticle platform. So for example, DNA, of course, in itself, by definition, is a biomolecule. And so just from that feature alone, we can actually decorate any other type of biomolecule essentially onto these nanoparticles. So for example, we can array them with other types of nucleic acids, so either other DNAs or RNA, even therapeutic RNAs, et cetera. Antibodies, proteins maybe, or any type of small molecule. So it's really a platform for when we're thinking of drug delivery, maybe we can essentially attach any of these onto these nanoparticles for cellular delivery. In addition to that, we're not limited to just attaching a single or a number of these. We can do the attachments in different combinations and also with a really high degree of spatial resolution. So for example, if we wanted to attach antibodies, we can actually do that in any number of these different patterns with different densities, et cetera. So that may become useful when we really want to control the exact spacing of things. We can do that down to the nanometer scale precision, which again isn't necessarily as easy with some of the other types of nanoparticles. And so taking all of these different features in mind, then we can maybe start imagining scenarios where this is a platform which can be useful for a number of different biomedical applications. So for example, maybe we can array sort of and attach different types of protein or antibody drugs onto these nanoparticles, which is only delivered to a cell when it reaches a particular cell of interest for true sort of cell type targeted drug delivery. Or maybe we can array different receptors onto the nanoparticles in a certain pattern or density, which activates particular cell states. Or even maybe we can create these nanostructures into various shapes, which resemble different cellular organelles or cellular components towards sort of an end goal of creating a true synthetic cell. So these are all really active areas of research, actually, in the field of DNA nanotechnology, which many groups are currently working on. And so I think, there's definitely a lot of exciting things to watch out for from this space. But one thing that, as a field as well, we've I don't want to say struggle with, but one limitation of sort of validating some of these methods is that even though they've all been validated at the proof of concept level, at the functional level, it's been surprisingly hard to actually visualize these nanostructures in situ in their cellular setting as they're carrying out those types of actions. So for example, typically what people do when they want to see the nanostructures in their cellular setting is that you can attach different fluorophores or dyes onto the nanostructures. For example, here they've attached about 7, I think, fluorophores onto this DN rectangle. And then you can introduce these fluorescently labeled nanostructures into cells and then visualize the fluorophores as a proxy for the location of the nanostructures themselves. But it's been found that this requires really is slightly insensitive in the sense that we really need to introduce a really high amount of the nanostructures just to enable the visualization themselves, which often may not, unfortunately, be super biologically relevant when we're thinking about the levels of concentrations which are actually present within an animal when we're carrying out these functions. And in terms of localization within an actual animal, which in this case you're seeing distribution of DNs lighting up with the yellow fluorescence, or I should say bioluminescence. These are typically done in animals at the really organ-wide scale, so it doesn't really allow us to look at cellular interactions of these nanostructures too much. So this is kind of the motivation originally that we had for developing these methods, is when we're thinking of really applying these for therapeutic different purposes in the lab, we didn't really have a good imaging method for us to validate our approaches. And so since it didn't exist, we wondered whether we can actually develop something that might be useful broadly to the field as well. And so that's kind of how OrigamiFISH was born. We wanted to really make a method which would allow us to image these DNA-based nanostructures universally within different cellular and tissue settings. And when we were thinking of this project, there were really a few features we really wanted the method to have to enable our downstream applications. So first of all, it should be ideally a really sensitive method in the sense that we should be able to detect very low concentrations of uptake, hopefully even single molecules of uptake, if that was really necessary for anything. Secondly, it should be able to be a high resolution method. So not only should we be able to image things at the single cell level, even within a tissue model system, but also hopefully at the subcellular level to really allow us to look at the spatial distribution of these nanostructures in situ. And lastly, something we were really motivated to do was to make this a label-free and universal approach, meaning that experimenters shouldn't need to think about their applications to modify the nanostructures anyway, just to enable the visualization. It should be a method that can be broadly applied to any sample after the uptake was already done without additional modifications to the DNs. So this is kind of what we aimed for the OrigamiFISH approach. And the idea behind it was actually very simple in conception. Essentially, we just wondered whether we can adapt another already existing and very popular approach called single molecule fluorescence in situ hybridization, or SMFISH for short, which is a fairly widely applied method in the biology field for imaging of RNAs within cells. So essentially how this method works is we have, of course, endogenous RNA, which is essentially just a single-stranded piece of nucleic acid. We can come in with the single-stranded DNA probes, which are fluorescently labeled, that because of sequence complementarity comes in and binds to these RNA molecules. And I'm just showing one probe here, but essentially in the actual method, you would have a chain of probes which are tiled all along the entirety of the mRNA, all of them being fluorescently labeled. And so that fluorescence accumulation essentially leads to an image like this, where you have a bright puncta, where each of the puncta within the cell is the location of a single RNA of that particular identity. So this is a very robust approach in the field already. And we thought, you know, RNA is a nucleic acid, DNA origamis are made of nucleic acids. Can we just adapt FISH towards detecting DNs and essentially establish our method as a sensitive approach that has all of the advantages of essentially FISH. So that's essentially the whole idea for OrigamiFISH in a nutshell. And just to go into a bit of detail of how we designed this then is essentially instead of targeting RNA molecules, we are targeting the scaffold DNA within essentially all different types of DNA origami designs. So the staple sequences, so the short oligonucleotides are the component that changes of the different shapes and designing different DNs. But the scaffold is essentially always the same and is always the M13 bacteriophage DNA. So by designing fish probes towards this universal scaffold, we wanted to aim our method at sort of a universal approach that works towards all DNs. And so we essentially just found places within the scaffold sequence, which is unique to the scaffold and doesn't cross bind to any other endogenous RNA. And then by designing and tiling probes all along the scaffold sequence, we can then initiate a fluorescent signal detection by using this secondary signal amplification strategy called hairpin chain reaction, or HCR. The details of this isn't super important. Essentially, wherever there's a probe that binds, this signal amplification strategy just initiates a chain of fluorescent polymers, which allows us to detect the location of the probes using fluorescence microscopy. So this is kind of the basic idea of OrigamiFISH in terms of the experimental workflow. And then this is, after doing OrigamiFISH, sort of what the images look like. So here you have the OrigamiFISH channel labeled in magenta, and then the nucleus of a single cell labeled here using blue. So each puncta, each magenta puncta you see here, in this case, is indicating a single nanostructure, which have been taken up by the cell, or a cluster of nanostructures. So right off the bat, we were kind of pleased to see that OrigamiFISH really, actually right from the beginning, allowed for these types of high-resolution imaging of both single cells and subcellular distribution of the DNs. And from these images, then we can do additional secondary types of analysis, which allow us to look at the distribution profiles of the DNs. So for example, we can segment out the OrigamiFISH signal to look at both the amount of puncta within a cell, to look at the general uptake levels within individual cells and across different cell types and across different structures. And so we can also look at things like intensity or the volume of the puncta to get different readouts for sort of dynamics and properties of these various DNs. Okay, so just to go into a little bit more detail on the actual protocol of OrigamiFISH, and just in case anyone is considering maybe or thinking of adapting some of these origami-based approaches, the protocol can essentially be divided into 3 different components, or at least I've divided it here into 3 different sort of phases. The first phase is really in terms of the material prep. So in terms of actually making the sample for doing the experiment. So this typically starts with making the actual DNA origami nanostructures, which is a fairly simple process. You essentially just add all of the DNA, so both the scaffold and the staples, into a single test tube, typically a PCR tube, thermo anneal that for a couple hours, and that really just automatically generates the DNs on its own. And then after purification, we can typically do an uptake experiment, which for cells typically consists of adding the nanostructures to the media of the cells that is being cultured. Or for animals, we just directly inject the nanostructures into, in our case, mice, but you can imagine that that can be tuned for different types of model organisms as well. So after the uptake, we allow that to happen for X amount of time. That's user defined, and we fix the cells down just like any other type of staining protocol. And then that kind of leads us to the second phase, which is the sample preparation. So essentially, this is the one part that is slightly different from regular fish protocols, which we've optimized specifically for these nanostructures, where we first fix the cells with PFA just to cross-link the nanostructures as well as other biomolecules to the sample. And then we go through a permeabilization step where we essentially use methanol or a combination of detergents to permeabilize the cell membrane to allow both the probes as well as different reagents to get into the cell. And then we also adapted a denaturation step, which is fairly critical for OrigamiFISH, because for the probes to get in, you essentially need to attack a single-stranded piece of nucleic acid, whereas, of course, the nanostructures are intact, double-stranded. And so we adapted a formamide and heat denaturation-assisted denaturation step to essentially release all of the staple strands from the scaffold to give us that nice sort of single-stranded scaffold, which can be attached to for probe hybridization. And then in terms of the protocol for OrigamiFISH, that part is essentially exactly the same as any other type of fish protocol. We go in with the overnight hybridization step, where we just add the DNA probes in a hybridization solution overnight. And then after that, we add in our HCR hairpins, which are commercially purchased, which all that does is, again, it just generates that fluorescent polymer chain, which allows for downstream imaging. So this is the entire protocol for OrigamiFISH, which from beginning to end is about a day. So not too intensive in terms of the experimental procedures. Okay, so now that we know that OrigamiFISH actually works, we really wanted to validate this approach as something hopefully that other people would also want to adapt. And so we thought of, sort of if we were users which were looking for techniques, what would we be looking for in terms of the imaging approach? And the first thing we thought of, and I think a really important aspect of the approach, is we really wanted to validate the sensitivity of the method, because that was a main limitation of previous fluorophore labeling approaches. And so I guess the ultimate sensitivity really is to develop this as a single molecule approach, right? So even if a single molecule of nanostructure got into a cell, we should still be able to visualize it robustly. And so that's the sort of one of the first things we wanted to do. This actually turned out to be a little more challenging than we expected, not because the method didn't work well, but because we didn't actually have a sample which allowed us to image single molecules of nanostructures within a cell. And that's because nanostructures get in, of course, through endocytosis. And so that's why these puncta that you see are so large, is because these are essentially the really large clusters of nanostructures which have gotten entrapped within endosomal or lysosomal compartments. And so before we could detect single molecules of DNs, we actually needed a model system first for making that happen. And so we ended up doing that through a collaboration with Dr. Yusen's lab, who are experts in sort of micro-injecting really small volumes of liquid or different solutions into cells. So essentially, our idea was that if we can inject really, really, really low amounts and numbers of DNs directly into the cytoplasm of the cell and then fix the cells right away, we can escape the endosomal fate of these DNs and then actually look at single molecules of nanostructures. So that's kind of what you see in this video here. You see the needle, so a glass capillary essentially being injected within the cell and then a pulse of the liquid being dispersed out. So that's just a really low volume of low numbers of DNs, which are physically being introduced to the cell. And then after this process, we just fixed the cells right away and then proceeded on with the regular OrigamiFISH protocol. And one thing I didn't mention is that we also injected at the same time a GFP solution just to know which cells exactly are injected. So here you see the GFP staining of a single cell that was injected with our DNs. And then after OrigamiFISH, we did thankfully see really clear signals still for the OrigamiFISH. Similar to what we would have expected, the spot sizes for the origami for staining after microinjection was much, much smaller compared to regular uptake, suggesting that these are no longer nanostructures which are entrapped within endosomal compartments and likely more to be single nanostructures which are actually within the cell. And then just to validate this further, we do know that regular RNA single molecule fish is a single molecule detection strategy. So we also compare the spot sizes between our microinjected OrigamiFISH signal with regular RNA signal and found that the sizes were also quantitatively and also visibly fairly comparable, again, suggesting that these are single nanostructures, which we were still able to visualize and confirming that OrigamiFISH is a single molecule imaging method. Okay, yeah, so that's kind of the concentration sensitivity of the method. The other type of sensitivity we also wanted to demonstrate was the temporal sensitivity. So can we actually visualize, and how early can we visualize the uptake of these nanostructures? And the reason we wanted to go after this problem was because currently in the field, we noticed that there was actually a really long gap between when people were introducing the nanostructures and when they were visualizing them. So typically the process is that you add the DNs to the cells and then you wait anywhere from 4 to 8 hours before actually imaging them, because that just is the time that the fluorophores need to accumulate within the cell. That seemed a little limiting to us because a lot, of course, could have happened within that 4 hours. And also endocytosis really occurs on the order of minutes. So we felt a lot of biology could have been missed by waiting for this amount of time. Sorry. And so I'll just go back slightly. And so what we wanted to do was to, again, introduce the DNs, but to figure out exactly how long we had to wait before OrigamiFISH actually started to work in a similar time course. So again, the experiment was fairly easy. Essentially, we just added the DNs to the cells and we waited for a time course of anywhere from, we went a little ambitious with this. We didn't even wait for the full 4 hours. We ended the time course at 30 minutes and we did anywhere from 1 minute to 30 minutes. And we were actually pleased to see that even at the 1 minute point after uptake, we were already able to visualize fairly clearly robust staining for OrigamiFISH at the early time point of the time course. And what was interesting to us as well was that we saw a somewhat membrane resembling labeling at these early uptake time points. So early on, we saw sort of a ring around the cell, which is suggesting, and we confirmed with membrane labeling as well, that these might be membrane absorbed nanostructures or cells which are actively being maybe uptaken within the cell. And over time, as you go towards more towards the later points of the 30 minutes, you see both an accumulation within the intracellular space of the origami, suggesting that the cells have actively taken in a lot of the nanostructures, and then less of these membrane adsorbents, which we also sort of quantified to a certain extent at the bottom here by differentially looking at these membrane spots versus cytosolic spots to try to get a readout for the dynamics of uptake for different nanostructures. And we did see that these dynamics were different depending on the shape as well, which would be logical as well based on biology that would be occurring there. Yeah, so this experiment was really just suggesting to us that we also had that temporal sensitivity. So we're able to visualize these nanostructures down to the second or a minute, at least, time scale to really look at that early phase of nanostructure uptake with this method. Okay, so as the last sort of validation of our approach, one more thing we wanted to make sure OrigamiFISH was able to do was to actually be able to differentiate between intact versus degraded nanostructures. And this is a question that often comes up typically in the question period is how are these nanostructures actually intact when you have so much nucleases within the cell? And one thing I haven't mentioned so far is that we do protect the nanostructures from degradation. In this case, using this K10 PEG coating, so a charge-based coating which attaches PEG onto the nanostructures, which have been demonstrated by other groups in the past to enable sort of protect the nanostructures from degradation. But we don't really know for how long the DNs are actually intact after cellular uptake because we didn't have a great method to visualize that in the past. And so we wondered whether we can sort of apply OrigamiFISH towards this application. And how we pitched to do this then is we essentially made a new design for these nanostructures in the form of a DNA rectangle where we labeled 4 different components of the DN using 4 different colors. The idea here is that if a nanostructure is fully intact, we would expect all 4 components and therefore all 4 colors to fully colocalize. But if they're no longer colocalized within a cell, that's probably suggesting that there's some degradation which has already occurred. And exactly how we did this is essentially we used actually also 4 different approaches to label the DNs to also get a readout for what the best approach was. So we either attached fluorophores directly onto the scaffold of the DN, or we had secondary exchange strands, which sort of acted as a handle, which added a fluorophore, but was and using a secondary approach essentially. So these are the OrigamiFISH independent methods, which are completely fluorophore labelings. And then we also attached or added 2 different methods for doing OrigamiFISH. So either using probes that are already described, which targets the scaffold strand, or we also added sort of secondary exchange handles, which from the staples themselves, which could also initiate OrigamiFISH. So as a staples initiated OrigamiFISH, if you will. So essentially for different colors that label for completely different aspects of the nanostructures. And then we did a similar uptake experiment where we just added the nanostructures at 2 different concentrations. So you see a high concentration at the left here, and then a lower concentration at the right here. And the 4 different colors just indicate the 4 different methods for labeling the DN. So we did see at the high concentration uptake that there was fairly good co-localization of the different colors, but it wasn't super informative just because they're so the signal intensity was so high that just by random chance that could have led to the co-localization anyways. What was more interesting was the lower uptake concentration. So at the 10 picomolar scale, we did start to see a lot of examples, and hopefully you can see a little better at the zoomed in image here. There are definitely spots which are co-localized for the 4 different colors, but more often than not, there were example spots, for example, here and here, which was only positive for 1 signal. So this is about 30 minutes after uptake. So this is suggesting that even in that shorter time scale, we are already seeing degradation, presumably, of these nanostructures, and really just highlighting the need for anyone developing these nanostructures, including ourselves, of the importance of really validating the integrity of these on a case-by-case basis as well, and that hopefully this might be a useful method for doing that. Right, so that's kind of the validation of the method that we did in a cellular setting. We also really wanted to extend, and this was one of our initial motivations, was to extend the method to different tissue model systems, which is sort of aligns with some of the downstream applications we had in mind in the lab at the time. So here I'm just showing an example image. So essentially we adapted the method then from the cell protocols to also different types of in vivo protocols, which actually didn't require a lot of modifications. Essentially, just instead of processing the cells, we just sectioned the tissue instead, and then did almost exactly the same OrigamiFISH protocol on the tissue sections. In this case, I'm showing a mouse lymph node. So we introduced the DNs intramuscularly, so similar to any type of vaccine application. Because we were trying to develop them for vaccines. And then we dissected out the mouse lymph node in this case and processed it for OrigamiFISH to look at the distributions. And we were happy to see that we were able to see, and so the OrigamiFISH signal is again in magenta here. So we were able to see individual single cells light up and also individual punctas within those single cells, which are corresponding to the locations of the DNs. And just as a further proof of concept, we did also make the protocol adaptable for immunohistochemistry. So we co-stain here with a marker for macrophages within the lymph node using F480. So in this case, you do see a large amount of co-localization with cells which are positive for this marker, suggesting that, and OrigamiFISH, suggesting that the DNs are likely really being eaten up by these macrophages, which also aligns with our biological understanding of nanoparticle uptake within the lymph node system or within the lymphoid system. We didn't really stop there. We wanted to make sure this was really a versatile approach that can be applied to different model systems. So we did apply this first, again, to the lymph node, but we also did it to the liver, which I'm not showing here, as well as the mouse brain in the form of mouse brain slices in this case. And then just to make sure this is actually also something that can work across different species as well, we also applied it to human brain organoids in this case, where you do also see robust robust uptake and visualization of the nanostructures, again in magenta here. To further, we really wanted to sort of test the limits of where we can go with this. So for the organoids, because it's a 3D model system, we also adapted a fully sort of whole mount staining method here for the organoids, where we essentially stain the entire organoid, which is approximately 1 to 2 millimeters in diameter in any direction. And with integrated tissue clearing and light sheet microscopy, essentially we can process the entire sample for OrigamiFISH imaging, and also image through the entire organoid to really look at the full distribution profile within the sample. And interestingly here, we found that the OrigamiFISH actually worked fairly robustly, but DAPI was the rate limiting step. So you might have seen that there was kind of a empty space within the center. That's actually because DAPI didn't penetrate very far into the center, but OrigamiFISH were able to be fairly robustly detected in this light sheet experiment. Yeah, so that's kind of the end of my presentation. At the end here, just a little bit of summary. Hopefully I've told you about our method, which is called OrigamiFISH, which really allows for this label-free and universal imaging of DNA-based nanostructures within cells and within tissues, and that we're able to image DNA uptake dynamics fairly sensitively, both in terms of the concentration as well as the temporal resolution. And that we also adapted it for both detecting the stability and instability of DNs, as well as made it compatible for various in vivo tissue model systems. Just as a slight pitch at the end, we do have a detailed protocol on protocols.io in case anyone is interested in or curious more about the protocol. And we're, of course, happy to answer and help with any type of troubleshooting as needed. Yeah. At the end, I do have a lot of people to thank for helping make this work possible. So I'll just take a little bit of time for that. So this work was done in my previous lab at the University of Toronto, as I mentioned. So I will thank my previous advisor, Dr. Leo Cho, for his support of this work, as well as in particular Travis and Megan in the lab, who helped with a lot of the experimental work of the work that I just described, as well as our many collaborators who really made all of the in vivo work possible using model systems that we didn't necessarily have a lot of expertise in. So all of the brain work was done in collaboration with Dr. Jinping Jia's lab, and specifically with Haiwan there, who helped us with the brain cultures. And then the microinjection work, I think I mentioned, was done by Wen Tian and Dr. Yu Sun's lab at the University of Toronto. And then all of the organoids were made and generated by Afrin and Dr. Reen Lee and Julia Mufat's lab, also at SIGKITS, and also to our funding sources as well. I was also supported by a Prime Fellowship for the bulk of this work. Yeah, and I'd be happy to take questions if there are any at the end.
Excellent. Thank you. That was a really beautiful talk. Thank you so much. And there are some questions in the Q&A and chat. So the first one, I think, addresses DN structures, and it asks, what actually mediates the structural rearrangement? I presume that is how the DNA and the hairpins work together to form different structures, if that is how I interpret that.
I see. Yeah, so for the actual making of the nanostructures is purely based on the sequence. So essentially, so the protocol is that we add the scaffold and staples together in a tube, and we do a thermal annealing step. So we raise the temperature, drop it slowly, and that heat essentially gives it the energy to, I think of it as they're just moving around in the test tube, trying to find their matches in terms of their partners based on the sequence. And once they do, they're designed in such a way where there's force being generated. So there's zigzags with the staples as well, where they're being pinched. So the long strand is being pinched. And once they're pinched at all sides, they essentially form into their designed shapes, which can then be purified out. There is actually also other ways to program. So the nanostructures, once they're formed, can additionally be programmed to morph into other shapes. So I wasn't sure if that might also be the question, but essentially all of that is done completely automatically using just the sequence alone.
That's really cool. Thank you so much. Rishit asks, since OrigamiFISH is sensitive to low concentrations, are there situations where this ultra-sensitivity might lead to false positives or signal saturation? And how would you mitigate that?
Yeah, so that's the one good thing about adapting a method which is already established, is people have really extensively validated the sensitivity and specificity of fish. We do have negative control images for all fur samples to show that we don't really see much signal in the negative control settings. There are, of course, a few puncta, maybe say 1 or 2 puncta per cell that light up, but it's in most cases, at least all cases that we've seen, it's really easy to differentiate between the positive and the negative. Essentially, it's exactly the same as fish in terms of the specificity of the method.
Thank you. Another question is, have you tried different organisms other than mice and human organoids? This person specifically works with C. elegans and is wondering if you've tried, or if you know of anyone who's tried working with C. elegans, since the cuticle is hard to penetrate.
Right. Yeah, I have not. Well, I have done some work with C. elegans, but not specifically for this. I think it would be really interesting if anyone did want to try. I imagine, yeah, as you mentioned, the cuticle. So I imagine you could either just micro inject it in, or maybe have the C. elegans eat somehow the DNs, but we haven't personally tried it ourselves. Yeah.
There's a question about resolution. What is the best resolution that OrigamiFISH can achieve?
Yeah, so I'm going to
Presume that's
assume we're
spatial.
talking about, sorry. I'm going to assume we're talking about the microscopy resolution. It's the same limitations as confocal, whatever imaging platform you're using, essentially. So in our case, we're using confocal microscopy. So we're talking about a 200 to 300 nanometer lateral resolution and probably 500, give or take, axial resolution. But we did think about developing this further as maybe extending it as a super resolution method, because you can imagine that the handles can easily be made to do that, but we haven't actually tried it ourselves. Yeah.
There's another question by Rishit. I hope I pronounced that right. Regarding probe penetration and signal uniformity. So the images of the mouse brain tissues are really impressive, especially the resolution of the single cells. However, tissue like brain or tumors have often dense extracellular matrix. So how does that affect probe penetration and signal uniformity?
Yeah, so those experiments, for also other reasons, were actually done on live cultures of brain slices, also to avoid some of the penetration issue. So I guess there's 2 questions. One for DM penetration. We did see penetration limitations in the organoid setting where it only went in so far, which is consistent with literature as well. So that is definitely a limitation. I guess you can maybe try to do it for longer, maybe, or use pressure to push it in, but we haven't tried it ourselves. In terms of probe penetration, these are almost always done on slices of tissue. So they're either cryosectioned or vibratome sectioned to 20 micron to 100 micron. So we don't see probe penetration issues at that level, which is consistent with FISH protocols as well. If you wanted to do it for longer, I think there's other groups who have just let it incubate for longer, and that's worked well for them. But again, we haven't tried that too much ourselves.
Thank you. There's a question regarding application of the technology. How can this technology be applied in, for example, neuroprotection? Can it be used in HT22 cells, for example?
I'm not too familiar with HC22 cells. I imagine any application, if there are other applications of nanoparticle systems for neuro protection, that maybe this can similarly be applied. I think as long as there's nanostructures in there, we should be able to detect them with OrigamiFISH. But in terms of the applications, I think that probably will be a case-by-case basis. Yeah.
Thanks. There's a question regarding DNs. DNs tag to be directed to specific compartments.
Yeah, so that's one of the
Can
first
they
things I
be?
OrigamiFISH as an application actually in the original. So people have done that, and we thought about maybe putting a nuclear localization sequence, maybe, or a membrane targeting sequence to put it into different compartments. That has definitely been done to, for example, target receptors to the membrane of cells by other groups. So yeah, it's definitely possible. You just have to put a targeting moiety on there somehow.
Thank you so much. I think this is it regarding questions. Thank you so much again. This was a really beautiful talk. I really enjoyed it. Oh, one more. Yeah, there's a lot of thanks coming in now.